Bismillahirrahmanirrahim. Students, in your previous lectures, you were taught about acute and chronic inflammation. Now the question arises, after these inflammatory events, how does the tissue restores itself? And what are the various mechanisms and events that takes place during this restorative process? All these processes and events they will be covered in detail in the following chapter of healing and repair. So let's start with the learning objectives for today's lecture, which includes to differentiate between regeneration and repair, what are the different types of cells in body, and what are the embryonic stem cells and pluripotent stem cells. First, we'll start with the regeneration and repair. Any injury to cells and tissues, they will start a series of events that will contain the damage, and at the same time, healing process will also be initiated. So what is regeneration? Regeneration refers to the proliferation and or multiplication of cells and tissues in order to replace lost structures. For example, the growth of an amputated limb in amphibians. On the other hand, repair consists of a combination of regeneration as well as scar formation by the deposition of collagen. So this slide shows a major difference between regeneration and scar formation. In regeneration, there will be complete restoration of a tissue, and it requires the intact connective tissue scaffold. In some tissues, there lies a partial potential to regenerate. In case of partial damage to liver and kidney, these tissues or these organs have the limited capacity to regrow their lost parts. And this is known as the compensatory growth, which also comes under the process of regeneration. In case of epithelium and bone marrow, complete regeneration takes place. Epithelium and bone marrow, they are continuously being utilized and replaced by the newly formed epithelium and bone marrow tissue by the process of regeneration. However, in case of repair tissue, the tissues will heal, but with the formation of scar. And this occurs when extracellular matrix is also destroyed. For example, in case of permanent tissue like heart and skeletal muscles, healing will take place but with the formation of scar. These tissues, they do not have the potential to regenerate once they are lost. So this slide shows a brief overview of healing response after an injury. On left side of this slide, you can see that after an acute injury, healing took place by complete regeneration, in which tissue is completely restored. And on the right side of the same slide, you can see that after acute injury, repair occurs with the formation of scar tissue. In case of the chronic injuries, healing takes place by formation of scar tissue and fibrosis. This slide shows the importance of intact cellular matrix after an injury, and the role of extracellular matrix in regeneration and repair. Whenever injury to a tissue causes loss of cellular matrix, the damage will be repaired by fibrosis. However, in case of intact cellular matrix, the tissue will be regenerated as a whole. So you can understand how the cellular matrix and how much it is important for a tissue to either to regenerate or to give rise to a fibrous tissue. So this slide shows how the cell populations are being regulated. The number and the proliferations within these cells or the tissues, they are maintained by increased and decreased rates of the stem cell input, cellular deaths, which occur by the process of apoptosis, and changes in the rates of the multiplication or proliferation. Coming towards the types of the cells in body, the tissues of the body, they are broadly categorized into three groups on the basis of their proliferative activity. The first one is continuously dividing or labile tissues. Second one is quiescent or stable tissues. And the third one is non-dividing or permanent tissues. So as the name indicates, the continuously dividing tissues are those tissues which do not require a stimuli to undergo division. These tissues, they continuously are being utilized and they are continuously being replenished and replaced by the newly formed cells and tissues. For example, surface epithelium of the skin, oral cavity, vagina, and cervix, lining mucosa of all the excretory ducts of glands, for example, the salivary glands, pancreas, biliary duct, columnar epithelium of the GIT and uterus, they are continuously being replaced. The transitional epithelium of the urinary tract and the cells of the bone marrow and hematopoietic tissues, all of these cells and tissues, they are continuously being replaced. Quiescent tissue. The quiescent tissues, they normally have a low level of replication. These cells, they usually undergo rapid division whenever they receive a stimuli, and in order to do so, complete regeneration or reconstitution of a tissue is attained. In this category, there are parenchymal cells of the liver, kidney, and pancreas, mesenchymal cells such as fibroblast and smooth muscle cells, blood vessel cells, lining cells, the endothelial cells, lymphocytes, and other leukocytes. Non-dividing cells. Non-dividing tissues, they contain the cell. Once they leave the cell cycle, they cannot go under mitotic division in postnatal life. For example, neurons, skeletal muscle cells, and cardiac muscle cells. Stem cells. The stem cells, they are characterized by their self-renewal properties, like they renew themselves, and they have the capacity to generate a different cell lineages. Embryonic stem cells. Embryonic stem cells are actually the pluripotent stem cells. They can be differentiated into different lineages, and they are isolated from inner mass of the blastocyst. The embryonic stem cells, they help in production of the human model diseases by knockout mice, that is inactivation or deletion of a gene from cultured embryonic stem cells, thus implanted these cells into blastocyst, or knock-in mice, which includes the mutated DNA sequences they replaces the endogenous sequences. Embryonic stem cells, they potentially repopulate the damaged organs. So this slide shows stem cell generation and differentiation. As you can see in the slide, the zygote is formed by the union of the sperm and egg, and it divides to form a blastocyst. The inner cell mass of the blastocyst will form the embryo. Cell of the inner cell mass, called as the embryonic stem cells, they are maintained in cultures, and these cells can be induced to differentiate into cells of the multiple lineages. This slide basically shows stem cell niches in different tissues. In photograph A, this shows stem cells which are located in bulge area of the hair follicle. In photograph B, stem cells within the intestine, they are located near the base of the crypt, above Paneth cells. And these stem cells are depicted in blue color. In photograph C, liver stem cells, they are situated in canals of the herring, which connect bile ductules and liver cells. And in picture D, corneal stem cells are located in the limbus region, and these stem cells, they are shown by blue color. And these are located between the conjunctiva and cornea. Induced pluripotent stem cells. These are also named as iPS cells. The iPS cells, they have been generated by reprogramming the adult differentiated cells through transduction of genes which encode these embryonic stem cell transcription factor. This has to be differentiated, and this has to be distinguished from generated pluripotent stem cells from adult differentiated cells by transferring their nucleus to a enucleated ovocyte. Such nuclear transfer techniques are notorious for bringing about the mutations. However, these induced pluripotent stem cells, they generate all three germ layers, and thus they can be genetically manipulated. These iPS cells, they can be useful for repairing the damaged organs and tissues and rectify the congenital defective organs or disorders. So this figure shows the stem cell therapy using the embryonic stem cells and induced pluripotent stem cells. On the left side, cells nuclei from a patient is introduced into enucleated oocyte. The oocyte will thus form the blastocyst, and thus these cells will be from the donor patients. In this way, embryonic stem cells can be obtained. If you see on the right side of this slide, you can appreciate that the patient cells, they are cultured, and induced pluripotent stem cells are generated in this way. Both the embryonic stem cells and induced pluripotent stem cells, they both are capable of differentiating into various types of cells. So By this therapy, the scientists and the researchers, they want to generate the damaged organs or to correct a genetic defect without causing transplant rejections. However, problem with this therapy is that cells DNA material, when it is taken out and is cultured, then mutation arises. If this problem is rectified, a huge breakthrough is expected in science. And this could, by achieving this rectification, a number of genetic disorders and damaged organs can be fixed. So that's all for today. Thank you so much.